So what is the pathophysiology of Guillain-Barré syndrome? So we talked about it being an autoimmune demyelinating condition. So it involves the immune system. So it's actually an immune-mediated process leading to neuropathy. And this immune-mediated process occurs after an infection, and it appears to be related to what we call molecular mimicry. So there's something that the bacteria or the virus contains. There's certain bacterial or viral proteins that look like proteins in our body, in our nervous system. And this leads to the production of ganglioside antibodies. So these ganglioside antibodies are the cause of Guillain-Barré syndrome. And an example of this molecular mimicry can be found with Campylobacter jejuni. So on the outer membrane of Campylobacter jejuni, there's a lipopolysaccharide that actually resembles gangliosides of human peripheral nerve membranes. So very interesting. The lipopolysaccharide looks like a ganglioside in our own bodies, in our peripheral nerve membranes. So our body recognizes this lipopolysaccharide and makes antibodies against it. However, those antibodies are targeting something that looks like gangliosides of our peripheral nerve membranes. So our own antibodies start attacking our own peripheral nerve membranes. And it doesn't happen with everybody who gets Campylobacter jejuni infection. So there's obviously some other parts of the pathophysiology that are missing, but this is what has been discovered so far. So on a peripheral nerve membrane, at one end we've got a neuromuscular junction, the other end we have the dendrites, and then in between the myelin sheath, so these would be Schwann cells in the peripheral nerve membrane, and in between each Schwann cell that makes up the myelin sheath is nodes of Ranvier. And there are certain ganglioside antibodies that are produced. One of them is known as anti-GD1A. An anti-GD1A antibody acts to actually attack the nodes of Ranvier. So this specific type of antibody, the specific type of ganglioside antibody, actually attacks nodes of Ranvier. And other antibodies that have been found are antibodies known as anti-GM1 and anti-GQ1B. These antibodies are more likely to attack the neuromuscular junction. So as you can see, even though we've got antibodies against gangliosides, they are still attacking different parts of the peripheral nerve. So it can lead to a difference in certain symptomatology. So we can see a different clinical picture depending on which antibodies are produced. And in addition to these ganglioside antibodies binding to different parts of the peripheral nerve, we also get complement cascade activation.